Good morning, everyone. Welcome to the Clinical Sciences Plenary session. Thank you for making it early this morning. If you can have a seat and we can get started, and then we can give Jonathan all the time that he needs, and maybe we can even get some questions afterwards. Thank you very much. So I'm Lena Sergitis. I'm one of the co-chairs of the clinical session, the clinical track. And along with Dr. Mike Silverman, my co-chair, I welcome you to the session. And I'm going to pass it on to Mike to introduce our speaker.
Good morning, everyone. So it's my great pleasure to introduce Dr. Angel. He's the head of the Division of Infectious Diseases at the Ottawa Hospital and the University of Ottawa. He's a clinician scientist, and his research interests range from basic mechanisms of how HIV influences the immune system through to clinical trials of new antivirals and immunologic therapies. A major focus of Dr. Angel's research is looking at novel ways to kill HIV-infected cells with a goal to finding a cure for HIV. Dr. Angel has served as the president of the Canadian Association for HIV Research and chair of the CIHR HIV/AIDS Research Advisory Committee. And welcome, Dr. Angel. Welcome to London.
Okay, thanks, Michael and Lena, very much for the introduction. And I want to thank Eric and Carmen as co-chairs of the conference. I'm not sure who the invitation came from, but anyone that came from, thank you very much. So I'm very happy to be here. So, CAHR's kind of my family. I think I've been coming to CAHR every year since 96-ish. I think I was involved with the recruitment of Andrew and Aaron, who run CAHR, who still run CAHR. So I've been involved and still talk to them regularly. And I've played in the CAHR Cup every year since its inception in Winnipeg, I think in 2018. A little disappointed Eric didn't play this year in his hometown, but Dylan, his grad student, kind of did his lab in the labs London proud, so thanks. So this is what I'm going to talk about. You see my title, it's very simple. I've tried to keep down the text. I don't have much text in here, so I can talk around the slides, and you just can listen to me. I guess you won't have to read much. For accreditation purposes, here are my conflicts of interest. I've got some learning objectives. This got put in because it's needed for accreditation. They're in the text, so I won't go through those. So, I took liberties, right, because I'm giving a talk, so I can speak on whatever take whatever approach you want. So I want to, just a brief introduction is talk about HIV cure research and how it came to be. And this is just a very few slides, and this is really my simple take on this. It's not necessarily in accurate chronologic order, but this is it. So this just represents the fact that today medical management of HIV is incredibly easy. It's a single tablet, multi-drug regimen, which is extremely well tolerated and works in just about everybody. Now we're getting into longer acting injectable therapies for more options, but the medical management of HIV is, there's been incredible advances, and it's perhaps not perfect, but it's nearing perfect. So that's point number one. And point number two is, you don't need to read the details here, but this is from some modeling from a study that was presented at CROI a couple years ago looking at life expectancy if you have HIV. So if you are diagnosed early and start treatment early, your life expectancy by this model is greater than someone without HIV infection. So there's a survival advantage of being diagnosed with HIV. This is clearly a model, and this has nothing to do with other aspects of health, but this is life expectancy only. So it was an interesting model and observation. So with these 2 points, I kind of came up with simple, well-tolerated therapy and, quotes, normal life expectancy, there's growing concern about lack of interest. And lack of interest for scientists means lack of funding, which is a real concern. Lack of interest in further understanding HIV pathogenesis. So if with treatment so good and people are going to live a long, healthy life, why do we need to understand how the virus causes the disease in the first place? That's just a kind of a concept. So, and then along came the case of Timothy Brown, which is the first case of HIV cure that was well published, well established, and now there's more similar cases or situations like this where individuals a curative HIV. So this led to, now with a demonstration that an HIV cure is possible, so some very creative scientists, I don't know who is at the, behind all of this, repurposed HIV pathogenesis work as cure research. That's very strategic, very opportunistic, and people kind of should be praised by doing this in order to leverage funds for what is, good discovery research. So things like establishment of reservoirs of HIV, characteristics of HIV integration, maintenance of latency, like along this, which were all pathogenesis work, now get termed cure research, and everybody's work now is looking for a cure for HIV. So that's very creative, and this has worked. So for the result of this, first resulted in big investments in the U.S. through the NIH and the Martin Delaney Collaboratories. $70 million was the first investment here, and that's grown. So that's 2011. And then the establishment of Towards a Cure. This is the IAS efforts, broad efforts. So to follow up on that, so this is what the CAHR did. So a couple years later, I just circled the dates. So there's the launch of funding cure research in Canada. So all of this is just how, kind of the creativity of the research community drove this. Maybe broader than the research community to drive this, but at least the research community drove this. So this was a contribution of $10 million for HIV cure research. And this was the announcement in 2014. So in the middle, that's Rona Ambrose. She was the Minister of Health at the time. So Eric Cohen was the principal investigator on one of the teams. He's back there. Hugo Sudens was a principal investigator on another team. That's Mark Ouellette to Rona Ambrose's right. He was the scientific director of the Institute of Infection Immunity. He was a big proponent, big advocate for good HIV research and HIV cure research, and did an outstanding job in his role there. And then Christopher Bunting is to his right from CanFAR. So this is just what CanCURE was. So that was the announcement of CanCURE, which is a Canadian team for HIV cure research. This is just from its website looking. There were cores and there were themes as a large team to support HIV cure research. That's the first iteration. So with some updated graphics from a few years later, this was renewed in 2019. Some more pictures to show really what CanCURE was, the model of CanCURE, how it went from basic discovery through to clinical trials. And as this is the clinical plenary, my focus will be clinical research. There's lots of basic science laboratory research going on in Canada. I'm not going to focus on much of that. I'll focus on the clinical trials. But this starts to provide some infrastructure for the establishment and conduct of those clinical trials. So that's CanCURE from 2019, and the additional infrastructure to support clinical trials in Canada in kind of collaboration with CanCURE is the CTN. So the CTN has been around since the early 80s, led by the last 10 years by Aslam Ennis, and now it's up for renewal, and it's been resubmitted. And Marina Klein, who is the new lead, and it's kind of driven by the CHR's call. It's kind of been reestablished and kind of reformatted, quite a bit different than the previous CTN. But what's embedded now within CTN, I just circled that in yellow, is there's a think tank for cure research. Before it was kind of encompassed within another one of what we had were cores. This is now a think tank dedicated cure research. So the CTN supports the infrastructure for clinical trials in all areas, but HIV cure research is one of them. So that's an important part of Canadian resource to support clinical research in HIV cure. So I'm going to go talk about a number of the cures. I'm going to highlight some of my friends that have done the work, and a number of you in the audience will be, I won't ask you to stand up, but I'll point you out as you do this. So the first, I think, clinical trial that was really done with the intent of looking for an HIV cure. I guess this is debatable which is really first. Looked at latency reversing agents. So these are molecules that cause HIV within the cells to come out of latency. So there's the potential to clear them from the body or from the bloodstream. some of the initial clinical work in HIV looked at latency reversal, and there's still ongoing work looking at such agents, but this is how it kind of started. So this kind of first started with a study of valproic acid. So it's in the top left corner, you'll see that's the CTN logo, because many of these, or all of these, were supported by the CTN in some way or another. So this is, and they're numbered through the CTN. So the CTN study, valproic acid and HIV. So looking at valproic acid, which is a latency reversal agent, to reducing the HIV reservoir. I'll get a bit back into that detail. So you'll see some common names through these, because the number of people that actually conduct these types of clinical trials is not a large number of people. So some of these are common. So Jean-Pierre is the lead. You'll see Jean-Pierre's name a lot. Cecile Tremblay's there. I'm in there. Rafik was in there while he lived in Canada. He's moved beyond us, but he's there in some of the early cure research. So this was the first one, and this is really just to show this. I'm not going to show results from all these studies, but this is to illustrate how Canada has started to get involved in HIV cure research. So then therapeutic vaccines is the next area of clinical trials that we've done. And so this is some of my work. So when I did this one, or we designed this one, it was in the late 2000s. We probably started in 2008, maybe, to start to design this trial. This is before the term cure was used. This is using what was referred to a therapeutic vaccine. The goal of this is to induce an immune response against the virus, hoping that would be able to control the virus once antiretroviral therapy was stopped. That's the principle of this. So it's a CTN study. It's a lower number than the previous one, because it started well before. But so this is not, at the time, this was never sold as cure research, but today this would be a typical kind of cure research study. And again, same name as myself, Jean-Pierre, Cecile, Rafik at the end, and other contributors there. And what we showed, and this provided some groundwork for further studies, not this approach itself, but we did show here, and it's not necessarily that easy to see. And with the vaccination before interrupting therapy, there was a slight delay in rebound viremia, and there was also a delay in restarting therapy. So these reached statistical significance, so it's nice to show some biological activity. They did not, the clinical significance is completely unknown, but it was not sort of as profound enough to continue to pursue this approach. But there's lots and lots of research going on in therapeutic vaccine work. Again, this is just to illustrate what we've done, and this cure research really predates the term cure research. And what's also good about these studies, and I'll show from a couple of these, lead to a lot of other work and a lot of other valuable work that will move forward. So looking at this study, we looked, the primary outcome of that was viral rebound, or time to viral rebound after stopping therapy or reinitiation of therapy, a clinically relevant outcome. We also looked at the size of the viral reservoir. That was with work with Cecile, the same investigators you see. We did this, so we looked at that as well. And it provides opportunity for other things. And the bottom one is something that is being picked up again now. So we did this with therapeutic vaccine study, and we wanted to know why people participate in a therapeutic vaccine study when the likelihood of any real benefit to them is small. People coming into these studies are not told that we're going to cure HIV. They're told this is a study. We're going to learn lots, hopefully, from this approach, and they'll contribute to this. And it became quite clear that altruism was the motivator for this. And now people are redoing all of this in the study, in the setting of cure research, why people want to participate in this. There's lots of reasons why people don't, why people do, why people don't, but it almost all comes down to altruism, which is an important observation we had, and people are repeating this today, which is nice to see. So the next one is another, would fall within, vaccines broadly. It's another study which was industry supported, but Jean-Pierre was one of the leads on this, and I participated in this as well, was looking at dendritic cell immunotherapy, where there's autologous dendritic cells, or cells were taken out, modified in the laboratory, and put back in. Again, I just want to illustrate kind of the complexity of some of these. Some of these are all investigator initiatives, some are industry supported, so the complexity of actually doing these. This was an interesting study. It didn't it pushed the field in understanding some aspects of doing these studies. It didn't really demonstrate a cure, which is fine, but it was really informative. And then what came out of this is, again, a paper published by Jean-Pierre looking at this, plus other studies I'd referred to in our therapeutic vaccine study, and he took this information out. There's about the safety of analytical treatment interruptions or structured treatment interruptions, or having study participants come off of antiretroviral therapy for a period of time, because it's a bit, some people feel it's controversial. There's potentially risk associated with this, but doing it in a controlled way, and as Jean-Pierre illustrated here and others have, this is a perfectly safe approach to conducting clinical trials. And so that leads to this. This is not work that we've done, but it kind of goes back to introduce all of this. I didn't put it as an introduction slide because I thought it would be not as interesting if I started, but I got to here. So there's three things I want to point out on this slide, just as this is a prompt to remind me. So the title of this study is Predicting Post-Treatment HIV Remission to Size the Viral Reservoir Matter. So there are a few things I want to point out here. So number one, they use the term post-treatment HIV remission. So when we first started doing this, people used the word cure and used the word cure alone. And then, I think people had different views about what cure meant. Some people would think that cure, understandably, people would mean that cure meant getting rid of every bit of virus in your body. Not everybody necessarily meant that. So then people adopted, scientists or whoever adopted sterilizing cure and functional cure. Sterilizing cure meaning getting rid of all the virus. Functional cure meaning perhaps, again, in a bit of a nebulous term, perhaps the ability of your body to control the virus without the need for medication, but people also would interpret that or use that meaning a little differently. So then what's been adopted in the vocabulary has evolved. Now post-treatment HIV remission is being used, and there will be continued evolution of this vocabulary. So it's more specific. It's not as simple as cure. Cure is an easy word to use. This is like 4 words, and unless you're kind of familiar with the area, post-treatment HIV remission is more cumbersome, but it's more accurate. So what the goal is to do is to have people that are on HIV therapy be able to stop HIV therapy without the need to restart therapy. So that's post-treatment HIV remission, using some cancer lingo in there. So that's number one. And cure will probably continue to be used. It's easy as historic, but for accuracy, it will kind of be eventually be lost from the vocabulary. Next, the size of the viral reservoir matter. I think that's an important question. We use the term reservoir, and conceptually, reservoir is an important thing. It's basically where the virus exists throughout the body, cells, tissues, when people are on antiretroviral therapy. People talk measuring the reservoir. We don't measure the reservoir. We measure things that are in the blood, aspects of the virus, genetic material of the virus, whether it's inducible or not inducible, whether it's intact virus or replication competent. We measure things in the blood that are a surrogate for the reservoir. So we do not measure the reservoir. Again, it's a lot easier to say we measure the reservoir. So I think people that do this understand it, so it's easier to communicate that we're measuring the reservoir. We absolutely do not measure the reservoir. We measure something that reflects, or is supposed to reflect, the reservoir. And we do that because we think having a smaller viral reservoir is good. We don't know that's the case. We think that's the case. We think that therapies that decrease the size of the reservoir is a good idea. Conceptually, it makes sense. We don't know if there's any clinical impact of that, but that's what we do, including myself. That's what we do. But I think what's important is that from this, and with respect, I just wanted to highlight, there's a good review. I didn't have time to put a slide, a nice review in Journal of Infectious Diseases that came out in March that Nicolas Chaumont wrote on measures of the viral reservoir, so you can read that article just to familiarize with how this is done, and there are lots of different ways, and it summarizes this work very, very well if you're interested. But the conclusion here that I underline that I think is very important is that consequently ATIs, or analytical treatment interruptions, are still the only way to assess the efficacy of new HIV curative interventions, just because measures of viral reservoir are not good predictors. Some studies, some are, some studies, some aren't. But the bottom line is this, and this means clinical trials that are going to show any clinical value become complex, because once you introduce an analytical treatment interruption, it's a larger, it's a more complex process. It's very valuable, but it just highlights the complexity of this. So that's where we are with that. So with respect to vaccines, I want to highlight some other work that's been done in Canada. So So the first one, top left, so it's Chris Tsoukas was doing HIV vaccine work long before probably many of you were born, and this is from the 90s, published in the 90s, and he was real lead in this area. So vaccine work or therapeutic HIV vaccine work in Canada has been going on for a long time. Another study here, and if you talk to Mario Skrewski or Colin Kovacs, their experience here, this is a CTN study of a DNA-based vaccine that never got going. I think the study got open but never enrolled. A lot of complexities here, so it was another CTN study. So just other investigators, Mario and Colin, involved in therapeutic vaccine studies. And the bottom left is just to highlight some stuff that's gone on in London. So that's Yang Kang, who's in London, and I think when Eric came here, he pushed Yang to publish some of the work he was doing. So this is a vaccine developed here in London, and this is a phase 1 study. We're all waiting for the phase 2 study, perhaps, to come out. But that's just to highlight some of the other work that's gone on in Canada and London in particular, since we're here in London. I'm sure Eric would be happy to answer any questions about this one. And then next is immune-based therapies, which is another area that a number of us have been doing some work in. And what has driven this most recently is this publication in Science. This is not from any of our labs, but this was a big kind of a breakthrough or a now debated, controversial observation, but at the time it was quite remarkable. The title, Sustained Virologic Control in SIV Macaques After Antiretroviral Therapy and Alpha-4 Beta-7 Antibody Therapy. So these are monkeys who were treated, they were infected with SIV rather, treated, and either they received control antibodies or alpha-4 beta-7 specific antibodies, interrupted therapy, and that curve kind of summarizes the main result. If you received the control, your viral load, the monkey's viral load rebounded to baseline. If you received alpha-4 beta-7, your viral load was controlled. That's pretty remarkable results. Others have tried to repeat it with varying success, but this prompted a lot of work in this area, and there's ongoing work with this area, and whether this is going to happen in adults or humans or not, it's unclear, but certainly this provides some foundation for additional investigation. So there's a lot of enthusiasm with this. So this resulted in another CTN study. It was a pilot study. One was first supported by the CTN called the Havarti study, and Bill Cameron can tell you why it's called the Havarti study. I'm sure he's told me about it, I don't remember. So Mickey McGinty, who was a fellow at the time with Bill, published the protocol, and it's a very simple protocol. People on antiretroviral therapy receive vedolizumab, which is the monoclonal antibody against alpha-4 beta-7, and then they interrupt therapy. And in this study design, there are 3 different doses of vedolizumab. And this is the primary finding. I can walk you through it, and Bill has all the details on this if you want more details. But the bottom line is with the lowest dose of vedolizumab, viral load rebounded to viral load set point or pre-antiretroviral therapy viral load and remained around that level. The next middle dose of vedolizumab rebounded but not to the pre-therapy viral load and stayed there a little bit, 1 log-ish below the viral load, below the baseline viral load until it started to rise. And where the blue line, that's the blue line, where the blue line ends, people restarted antiretroviral therapy. And the green line, which is the highest dose of vedolizumab, granted it's only an N of 1 right now, they didn't rebound high, and they've stayed with a low-ish viral load, about 1.5 logs below their pre-therapy viral load. So this is what appears to be some sustained activity of vedolizumab. And if you look at this time from, or if you look at this, looking at dose of vedolizumab versus time off of antiretroviral therapy, there's a dose response with a higher doses of vedolizumab leading to a longer time off antiretroviral therapy. So this is work that's ongoing and at our site trying to continue with this. So again, an interesting finding here. Other work that's done, so this is, again, highlighting more Jean-Pierre has done. This is a study, this is an industry-sponsored study, just looking at additional immunotherapies that have been used. So this is a PD-1 inhibitor that's owned by AbbVie, and it was done in a similar study, an ATI study, analytical treatment interruption study. Participants received the PD-1 inhibitor or placebo. They stopped therapy. And this is what Jean-Pierre has presented Recently, I think this was a late breaker at CROI or at EECS, it's presented a couple times, but you can see in the bottom graph, the participants that are on placebo, they had their viral, on placebo, the viral load rapidly rebounded, and those that received the PD-1 inhibitor, and a couple, there's a really profound delay in viral rebound, if at all. So this did clearly perturb the system. Some participants seem to benefit, and clearly different than placebo. And if I remember, you guys may not remember from the slide I showed from the therapeutic vaccine trial, this is almost identical, the delay in viral rebound, the mean delay in viral rebound or median, 21 days in placebo, 29 days with the intervention. So you can delay viral rebound here a week. Again, how important that is unclear, but it's quite remarkably similar to what we've shown before. So this has led to another study that a number of sites are participating in. Bill Cameron's the lead at our site, Jean-Pierre's the lead. There might be a couple other sites in Canada. So this is combining the PD-1 inhibitor from the slide I just showed you and a monoclonal antibody against alpha-4 beta-7, so a molecule similar to what I showed you in the Havarti study. So they're combining this and then going to look at an analytical treatment interruption. This is an ongoing study, and there are no data to show. But again, highlighting what's being done here and how some of the work that's being done in this country is kind of at the edge of clinical trials in HIV cure work. Another thing through CanCURE and through some serendipity, we've looked at other aspects of HIV cure research, and this kind of just fell in our lap, but we're kind of running with this. So it's a real interesting kind of newish field that not a lot of folks have been involved with, is research at the end of life. Using this as a broad term. So in the top left, we reported on it was a patient of mine who was undergoing MAID. I got a phone call from the MAID, that's medical assistance in death. I got a phone call from the physician that was overseeing the MAID, and she was a friend of mine, and she called me and says, this patient's being enrolled in MAID, and I'm going to be undergoing MAID next week. He says he really would like, if he can, contribute to the research or contribute to science. Is there any way in which he can? Which I thought was really an interesting concept that had never come across, my desk before. I'm not sure, this may have been the first patient I had that underwent MAID. So I said, let me see. I talked to lots of my friends in Ottawa and in Montreal to see if there's anything we could do with his desire to participate in research in any way. So there was a big effort over a relatively short period of time. His family was all on board, which was important because after someone undergoes MAID, you need, there's still family involved to know what to dictate what to do with the body. So we fortunately had a pathologist that was supportive. We had folks from Montreal, from Jean-Pierre's group, from Nicola's lab, Petronella's lab, Petronella and Kuda come over to Ottawa. They had the autopsy. We had to come into the hospital. He wanted to go MAID at home. Came to the hospital. All his tissues came to my laboratory, and the folks, graduate students, technicians, spent the weekend isolating cells and tissues in order to study, I use the term HIV reservoirs, but tissues throughout the body where HIV can be hiding, and to describe those. So this was the first one of those. I'll show you in the next slide a little bit reference to what's come of that. So that paper just describes that opportunity of patient wanting to contribute to HIV research and our ability to do it, which was no small task. And then Cécile Tremblay and David Lessard in Montreal have kind of taken this a little further to look at, so this is some qualitative research, the willingness of older Canadians to participate in HIV research towards the end of life, part of which would be after death. So there's a ongoing study in Montreal looking at the kind of qualitative aspects, but also enrolling people in an autopsy study. Right now, the numbers are small. Other folks have been doing, Mary Ostrowski is doing this in Toronto. There's challenge that any one of us can talk about. In Toronto, Chris Power in in Edmonton has contributed to some of this as well, and Cecile Costinic in Montreal are kind of the leads. And so this led from the patient we had in Ottawa and the patient that Chris had in Edmonton. We supplied or provided Nicolas Chaumont's lab with lots of tissues. This is just a graphical abstract here when the folks in his lab described the virus throughout the body and all the tissues. A lot of sophisticated work doing this. And that's the title. You can read the title. But what this allows us to do is to study virus in places that you can't do in people that are alive, right? So right now we just can measure things in blood, but this opportunity of having people that want to contribute after they die, it really provides a kind of a unique opportunity to study virus in the brain, in the lungs, every organ, places we're unable to, which if you're going to cure HIV, you really need to understand, I think, where the virus hides, not just in the bloodstream. So I'm going to talk very little. I said I was going to talk about clinical work, but I want to do this just to highlight some of the other people that are involved. Some of this preclinical work has the potential to move to clinical work, which is the goal of all of this. So this is work from Jerome Estequier. This is using a molecule. It's an anti-caspase inhibitor molecule, so it inhibits apoptosis. It's anti-caspase, so it inhibits the apoptotic pathway that they've used in macaques, SIV-infected macaques. So this just summarizes the results from this study, which leads to more studies. So perhaps the bottom right image is the more important one. So it looks at, this is a typical slide of viral rebound after stopping antiretroviral therapy. In the placebo group, the virus rebounds to the pre-therapy level, where in the treated group, the QVD treated group, you get about a 1 to 2 log decrease. So there is, again, some biological activity with this group, and then with this, you can design, if you're working with animals, you can design studies that will mimic what we will eventually do or ideally do in humans. So they've designed again, a therapeutic interruption trial. This is the same as like the number of the studies I've talked about. The monkeys get infected with SIV, they get treated with antiretroviral therapy, they get treated with QVD around the time of interruption, and you look at viral rebound here. And this is kind of summarized on the right. You get about a 1 log decrease in plasmaviremia if you received QVD versus you didn't. So these are very preliminary results. These haven't been published or looked into more detail. But this shows, again, a biological activity of this agent, which will prompt more work in this area, and then if and how and when this moves into humans, that all needs to be figured out. But this just, again, another demonstration of some of the work that we're doing. And then the last is just, because I'm here, I can just promote myself, so it gives me the opportunity about some of the work that we've been doing, and I'm going to go through this relatively quickly. It's a lot of work in a relatively short period of time, but I think it's fitting for this talk, and then showing where we are with support from particularly CanCURE in moving discovery through to clinical trials, I think, which is all what we'd like to do. So this is lots of work on one slide, but you don't have to read it all, but I'll walk through it. So we first, I work closely with a group in Ottawa that does cancer research, and they look at viruses that are able to kill cancer cells. And one of the reasons why these viruses kill cancer cells are because cancer cells are inherently different in a number of the proteins within the cells that protect them from being killed or make them susceptible to being killed. And we identified that some of the defects in some of the cancer cells are similar to some of the defects within HIV-infected cells and latently HIV-infected cells. That's the first one. So that was the first observation we made in order to move this forward. And then we looked at the viruses that kill cancer cells are actually able to kill latently HIV-infected cells. So we've done this in vitro in cell lines. We've done this in cells from participants that are infected with HIV, can kill their cells. We've actually done with support from Ali and Cecilia. We've looked at cells from the lungs obtained from people with well-treated HIV, that in primary cells that these viruses can kill HIV-infected cells. So we've done just about everything we can do in vitro. There's not much more moving this forward. We can enhance it with other molecules or modify the virus, but we've shown right now as far as we can, in all the models that we have in vitro, these viruses kill latently HIV-infected cells. So the next step is to go into animal models, and I think a lot of people talk about, oh, we'll just then we'll go into animal models, and the concept is fine, but actually doing it is complex. And this is just to show you that we've actually done it. This is the first time doing it, so this is work supported by, so this is using humanized mice. I'm not going to talk about these models. I talked about models of HIV in non-human primates. These are humanized mice that can be infected with HIV and treated, and uses a model of HIV. So Kathy Beeland and Ellie Haddad in Montreal have the resource that generates these mice. We supply them with tissues, actually from Ottawa, some fetal tissues, some thymic tissues, so that they're able to generate the mice. And then Tran Pham and Eric Cohen, with Eric, are the ones in Montreal that do the HIV infection. So I have a graduate student that has been going to Montreal to actually do this work. So just to show that this is not a concept, this is actually really happening, because there are dates on the bottom. So the mice we have been infected with HIV at week 6. So April 3rd, they started treatment with antiretroviral therapy, and then they will get injected. So the dates are there. So this is actually happening. So this is not theoretical. And this is just to show you. You don't have to believe me. You can see the data here that these mice are infected, right? So this is actually happening. So we are going to we're going to be looking at MG1 in these mice to see if this, and I have no idea if it's going to work or not, but this is how we just move forward with the science. And so that's just all I want to talk about. That's all the studies I want to show you. Just to summarize with this, so Canada's contribution to the global HIV cure effort has been substantial. However, there remains ample opportunity to engage in and contribute to this field of research, as you can probably imagine, because we still are pretty far away from actually, I think, having a cure. And this is my acknowledgements slide. There's lots of names here, people I've mentioned through the talk, and the organizations that have supported this. Can't list the numerous trainees, research staff, study participants, and their families as well needed to do this. And the two people I have in red are really the kind of the leads across the country that are doing real clinical HIV cure research. So these are, this is not something you do off the side of your desk. This is a commitment to your career. They're complex, time-consuming, very interesting, very challenging. But Bill Cameron and Jean-Pierre have really been the leads in Canada for doing these types of trials. So they get to have their names in red. So that means Bill has to be kind to me for at least the rest of the conference. And that's it. Thank you very much. Okay. Oh, you're welcome.
So actually, Jonathan did it in record time. So if we have any burning questions in the audience, we can take them. And it looks like we do.
Oh.
My question's not burning, but
Hi, Jessica.
hi, that was a really great overview. Thank you so much. I do a lot of work in East Africa, and we've been starting to think about what would structured treatment interruption studies look like, but then also what would a future with people in remission look like? And I'm wondering what you as a clinician think of, what would that look like in terms of constant monitoring of people who are in HIV remission? It's a little different than cancer, right? Because HIV is infectious, and if someone's in a relationship, a serodiscordant relationship, they may want to really know what their viral load is.
Yeah. So I think that depends on how we want to define remission. So it's often defined as no need for therapy. It's not defined by, or can be defined, but there's no specific definition by viral load. So if you become or allowed to become the equivalent of a long-term non-progressor, where you have virus but a stable CD4 count and everything else is stable, that's very different than if you're in remission and I think very different than in remission and have an undetectable viral load. Okay, so those things are probably very different although they may not be different in their natural history in an individual, they're likely very different in the ability of the person to transmit HIV. So first, I think it depends how you want to define remission, and I think we're not even there yet. And I think it's going to use a lot of judgment, and I don't have a specific answer because I think it will be tailored to what the remission is. So someone who's in remission has an undetectable viral load off therapy, just like we do for elite controllers, right? That's a model. And it varies. We'd like to see them maybe every 6 months. They don't like to be seen, right? So they're there's part of it is reinforcing on them how well they're doing, how healthy they're doing. They want, many of them, it's hard to generalize, but my experience, they want to avoid medical care. From our point of view, it's nice and reassuring and probably good care to monitor them. So that's going to be a balance. So there's what remission is, how you define remission, what you would want to do as a healthcare provider, and what the patient would want to do based on their situation. So it's hard for me to give you a specific answer because it really depends on the scenario. Is that okay?
Yeah.
Okay. Okay, good. Just for you.
Yeah. No other questions, then thanks, and we give this little gift that has the cure for HIV in it.
Thanks very much. Thanks.